All right, welcome back to part 3 of transcription. So by the end of this lesson, hopefully you will be able to list the functions of the 3 types of RNA polymerases in eukaryotes, describe the characteristics of eukaryotic promoter for a protein encoding gene, explain how general transcription factors in RNA polymerase assemble at the promoter and form an open complex, and compare and contrast the 2 possible mechanisms for transcriptional termination. So, many of the same features of gene transcription that we saw in bacteria are true in eukaryotes. However, what you need to remember is eukaryotes are much more complex. So they're larger, complex cells, meaning that they have multiple organelles. Um, this added complexity means that there are more genes that need to encode proteins, and that those genes need to be regulated in a particular fashion. If you step up from eukaryote to a multicellular eukaryote, that even makes things more complicated because now we need cells to be able to specialize, which will involve turning on or turning off particular genes. So you can see how eukaryotes need a slightly more complex process. So let's start diving in here. First of all, eukaryotes have multiple polymerases. So there are 3 that we are interested in here. So RNA polymerase 1, this is responsible for transcribing most of the ribosomal genes, this one exception here. RNA polymerase 2 is the one that we're going to be dealing with the most. This is responsible for all of the protein encoding structural genes. This is also involved in transcribing some things called snRNAs, which we haven't talked about yet, but these are involved in splicing, which we will talk about coming up soon. Finally, we have RNA polymerase 3. This transcribes all of the transfer RNAs, also that last remaining ribosomal RNA, and also a group of RNAs called microRNAs, which have an added functionality in gene regulation. We will come back to those later on. All right. All 3 of these polymerases structurally look very similar, and they're actually very similar to the bacterial RNA polymerase. So if, here's a picture of what a bacterial polymerase looks like. Here's a eukaryotic polymerase, so this bacterial, eukaryotic. They have the same overall shape. So we have a base wall that houses a catalytic site with a magnesium ion. There's this area called the bridge, and there's a pore for nucleotides to come through. There's a clamp. This has that thumb and finger thing that we see with DNA polymerase. But there's also added structures here. So here is something called the rudder, which breaks apart the RNA-DNA hybrid section to allow the RNA to come back out and the DNA to re-anneal. But overall, fairly similar. So the eukaryotic promoter sequence have a little bit more variety and are much more complex than what we see in bacteria. So eukaryotic genes have 2 major features. There is the core promoter and regulatory elements. Within the core promoter, we have this sequence here, TATAAA, or the TATA box. This is analogous to the -35, -10 sequences. there is also a transcriptional start site. So let's take a look at that. Here is the actual gene. Here is the +1 transcriptional start site. So when we make RNA, it will start there. Just upstream of that, we have the TATA box. And this part right here, these two in combination, are the core promoter. And with just a core promoter, you will get transcription of genes at a very low level. So let's also upstream, we have this section which has regulatory elements, but we will come back to that. Let's focus in on this core promoter first. So there are several proteins that come together, three main categories here. So we have the polymerase itself. We have a group of 5 proteins that we call general transcription factors. These are going to be different from something that we call regulatory transcription factors. So the general part of this word is important. These are involved in all genes. And then we have a protein complex called the mediator. So here we have the gene, we have the TATA box, we have transcription factor 2D that will bind to that sequence. Transcription factor 2B will recruit to B, so D recruits B. B will recruit the polymerase, which already has F bound to it. And once you get this clustering together, we can get transcription factor 2H and 2E coming together. So, H itself is a helicase, so that will help open up the complex. And it also helps activate the polymerase, has a carboxyl terminal domain on it that needs to be phosphorylated in order to activate it. Transcription factor 2H is involved in that process. Once that takes place, you will get the beginning of, you will go from a closed complex to an open complex. You'll get beginning of transcription. You can see that we now have phosphorylation that occurred down here. And at this point, D will separate from the rest of the elements. B, E, and H will fall off. And then the RNA polymerase is allowed to progress onto the elongation phase. So it'll continue forward, making a longer and longer gene. So for basal transcription, we need the RNA polymerase II and the 5 general transcription factors. The third component that we need is this large protein complex called the mediator. And there's many subunits, and it's kind of a mix and match scenario. There are different members for different genes when it comes to the mediator complex. So as it suggests, it mediates an interaction, and this interaction is between the RNA polymerase and all of the regulatory transcription factors. So these are not the general transcription factors that we see up here. These are involved in upregulating or downregulating that particular gene. Those transcription factors bind things called enhancers or silencers. Enhancers, you can guess that they enhance transcription. Silencers will block transcription. So the mediator is also involved in the phosphorylation of this carboxy terminal domain. It says here the mediator may phosphorylate it, and this all comes down to what is being included in this mediator complex. So again, it's complex and variable. Some of those variables have phosphorylation capacity. So let's come back to this idea of regulatory transcription factors. So upstream of our core promoter, we have a region from about -100 to -50, where which has DNA sequences that are called regulatory elements. To those regulatory elements bind proteins called regulatory transcription factors. They will come in and bind to those. And there are two general categories. So there are enhancers, they stimulate transcription, and silencers, which inhibit transcription. Now, we point out this region here as -100 to -50. And this is where we usually find at least some regulatory transcription factors and elements operating. But these regulatory elements and regulatory transcription factors can really exist anywhere. Some will be downstream, some can be thousands of base pairs away, some might be in introns. So there is a lot of variety of how that can happen, but some of the common ones will exist in this region upstream. So keep in mind that we have elements and factors. So the elements are DNA sequences. So this an example in the core promoter would be the TA box. But for the regulatory section, we have enhancers and silencers. So these are cis-acting elements. These are part of the DNA, the sequence itself. Trans-acting elements are those regulatory proteins that bind to the cis-acting elements. Now, eukaryotic messenger RNAs are heavily modified. So you've heard about introns and exons before. There is even more modifications that take place. One of this is there is a sequence that will be on the transcript that is known as a poly-A signal. That signal triggers that messenger RNA to be cleaved and to then flow off. Afterwards, you will get an attachment of a whole string of A's to the end of it. The transcription itself will terminate 500 to 2,000 base pairs downstream of this poly-A signal. So the RNA polymerase will just keep chugging on until it gets terminated. And again, there's a couple different models to figure this out. But it'll keep chugging along. The actual pre-messenger RNA will have been cleaved off and moved away. So let's take a look to see what this looks like. So here is a piece of DNA. Encoded in there is a sequence that we will call the poly-A signal sequence that will get transcribed into the messenger RNA. Shortly downstream of that, you will get a protein that binds to the poly-A signal sequence and cause cleavage to occur. Following that cleavage, one of a couple things seem to happen. Either it being cleaved destabilizes the entire complex and it just disassembles, or you can have an exonuclease that starts chewing up this unneeded piece of RNA that is still being transcribed. It might come up and, similar to Rho, make contact and knock it off. There's evidence for both of these, and they aren't actually mutually exclusive. So eukaryotic cells may use a combination of both of these techniques, or it could be that we are just not looking at it in the right way.